Have you ever heard that sharks can smell a drop of blood from a mile away? Research has recently shown that this is not true. It was discovered that they can only smell about as well as other fishes, effectively debunking this urban myth. Elasmobranchs include sharks, rays, and skates. It is important to understand the sensory biology of these species because it affects how they find mates, avoid predators, and find food. So, in essence, the keys to their survival. However, there are still large gaps in this knowledge. The general structure of the olfactory system includes an olfactory bulb and a capsule that houses the olfactory rosette. These rosettes are comprised of soft, squishy tissue that are stored in a rigid structure. Inside the rosettes are sheets of sensory epithelium called lamellae. Odorants are brought in with water, and they're detected by receptors on the lamellae. The size and shape of these organs can vary from species to species, but recent research has demonstrated that there's not a positive correlation between organ size and sensitivity. The size, shape, and number of lamellae do not necessarily provide an accurate picture of a species' ability to detect odorants. For example, the hammerhead shark has large rosettes, but it does not mean that it has a better sense of smell than other species. It is not yet fully understood why this variation exists. Why would rosettes occur in a variety of shapes and sizes if it doesn't impact odorant sensitivity? It has been hypothesized that it is related to the pattern of water flow throughout the system. My research aims to fill the gaps in this knowledge by investigating the diversity of rosette morphology. If you've ever had a CAT scan done at the doctor, you might be familiar with the CT machine. I use a smaller version of this called a micro CT scanner to quantify the morphology of these organs in a natural position within a fish's head. A CT scanner uses X-rays to create a reconstructed three-dimensional model of an object. Most of the existing data on rosettes has been collected on organs that have been dissected from a specimen's head. 3D reconstructions allow us to better understand the morphology of rosettes and the potential impacts of location and orientation within the head. Once we have these 3D models, we can use special software to investigate how water flows in varying olfactory rosettes. For example, organs that are longer and skinnier might have unique flow patterns when compared to organs that are short and wide. Together, morphology and flow could be optimized for odorant detection. In summary, these factors may help elucidate the question of why such an important sensory structure would exhibit drastic differences in morphology. The broader implications of this study include the development of bio-inspired chemical detection devices. Examining how the sensory organ of elasmobranch fishes functions can lead to improved sensing devices for diagnostic medicine and homeland security. We know that sharks can't smell a drop of blood from a mile away, but my research will answer more questions about how these sharks can smell.